Number 14. The scar beneath the Pacific. More than 10,900 meters of ocean stands above Challenger Deep, making it about 7,000 feet deeper than Mount Everest is tall. At the bottom, the pressure approaches 1,100 times that at sea level. The water is nearly freezing. Darkness is absolute. It should be one of the least explored and least inhabited places on Earth. Instead, it has become a changing archive of discoveries. The Mariana Trench stretches about 2,500 kilometers southeast of Guam, where the Pacific Plate bends and descends beneath the Philippine Sea Plate. The first recorded depth measurement came from the 1875 Challenger expedition. James Cameron reached the bottom alone in 2012. Victor Vescovo returned in 2019 with a vehicle built for repeated dives. Each descent revealed a different bottom, a different visitor, or a different limit. No expedition has seen exactly the same world twice. The place where measurement itself starts to fail is not a point on a map. It is a moving target buried beneath 36,000 feet of water. In the western Pacific, about 370 kilometers east of Guam, the seafloor folds into a long, narrow scar. This is not an isolated hole punched into the crust. It is the visible edge of a subduction zone, where the Pacific Plate bends downward and disappears beneath the Philippine Sea Plate. The trench is roughly 2,500 kilometers long. Challenger Deep occupies only a small section at its southern end. An expedition is not searching one neat cavity. It is entering a geological system larger than many countries and trying to identify its lowest depression from the surface. First sounding in 1875 gave the trench its name, but it could not give scientists a perfectly fixed bottom. A weighted line reached the seafloor at one location, under weather, current, and ship motion conditions that could not be completely controlled. The result was a depth measurement, not a final coordinate. Modern instruments changed the method, but not the underlying problem. Sonar maps the seafloor by sending sound downward and measuring its return. The calculation depends on the speed of sound through layers of cold, dense seawater. It depends on the ship's position, tides, gravity, and the reference level used to describe the ocean's surface. Two surveys can examine the same deep and produce slightly different maximum readings. That discrepancy is not a mistake sitting in the margin. It is part of the evidence. Depending on the survey method and reference datum, modern estimates place Challenger Deep at approximately 10,900 to 11,000 meters. NOAA gives a commonly cited figure of about 10,935 meters, or 35,876 feet. Other measurements have identified nearby depressions with subtly different depths. The difference may be only a few meters. At the bottom of the ocean, that is enough to change which point is called the deepest. On March 26, 2012, James Cameron descended alone inside Deep Sea Challenger. The vehicle was built to carry one person through a pressure regime capable of crushing ordinary equipment. Every cable, viewport, and control system had to survive the descent. Cameron was testing whether a human-operated machine could reach the point where the ocean presses hardest against the planet. Reached the bottom, but the visit was brief and the view was narrow. A camera sees one direction. A vehicle occupies one position. Sediment can cloud the water. Lights reveal only a small circle. The trench floor is not perfectly uniform. In 2019, Victor Vescovo returned in Limiting Factor, a vehicle designed for repeated dives. Multiple descents allowed researchers to inspect more than one section of Challenger Deep and compare observations under better controlled conditions. The returns did not produce one frozen image of the abyss. They produced a series of local scenes, sediment, slopes, scattered material, and measurements that had to be matched against a wider map. You are sitting inside a pressure sphere at the southern end of a trench that extends for thousands of kilometers. Above you, the water column is taller than Everest. Beneath you, the crust is being pulled into the mantle. There is no visible horizon, no natural light, and no obvious boundary announcing that you have reached the absolute bottom. Only an instrument reading says how far down you are. Even that reading carries a margin. The trench is chemically active as well as deep. Within the wider Mariana Trench Marine National Monument, NOAA, has documented unusual volcanic features, including liquid sulfur associated with the Daikoku submarine volcano. The region is not simply a pressure chamber at the edge of biology. It is a connected system of descending crust, volcanic heat, unusual chemistry, and sediments that collect whatever the ocean sends downward. Every expedition can return with a different answer to the same question. One dive measures a depression. Another crosses a neighboring basin. A vehicle records a biological trace. Another finds human debris. A third reaches a slightly lower patch of seafloor and changes the number by a few meters. The trench does not present a single scene waiting to be photographed. It presents fragments, each tied to a different coordinate, instrument, and moment. The deepest verified figure is about 10,935 meters, but the exact lowest spot is still a precision problem. Challenger Deep has several named depressions, and the deepest reading depends on how the seafloor is surveyed, corrected, and referenced. The scar is real. The number is not singular. Number 13. The rope that kept going. On March 23, 1875, a weighted line disappeared over the side of HMS Challenger, somewhere between Guam and Palau. It kept running out into the western Pacific, past every depth the crew had expected to find. No camera followed it. No sonar screen showed a descending mark. There was only rope, water, and the slow movement of a ship waiting for the lead to touch bottom. The sounding turned empty water into a destination. HMS Challenger had left Britain on December 21, 1872, carrying an idea that was still almost absurd. Convert a naval corvette into a floating laboratory and measure the oceans as if they were a new continent. The crew would record temperatures, dredge the seabed, collect animals, and lower instruments into water whose depths were mostly guesses. The expedition would last until 1876, crossing the world with scientists, sailors, boilers, specimen jars, and an enormous amount of line. On this March morning, one man's job became strangely simple. Lower the weight, watch the line, wait for it to stop. The sea gave no visual clue. The surface looked ordinary. The horizon was flat. Beneath the hull, however, the line continued downward through thousands of meters of water, carrying the ship's only direct message from the seafloor. A modern expedition would send an acoustic pulse and receive an electronic return in seconds. The Challenger had to feel its way into the dark. The line passed 1,000 fathoms, then 2,000, then more. A fathom is 6 feet. The old measurement was not a neat cable marked for convenience. It was a working line carrying a heavy lead through a column of seawater. Drag, stretch, current, and the ship's movement could all affect it. Every additional length made the result harder to trust. The farther the weight went, the more the ocean turned a measurement into an argument. The expedition had not come looking for a named abyss. It was trying to build the first global map of the deep ocean. The Mariana region was one station among hundreds. The number written in the log would have to survive questions about the instruments, the angle of the line, the weather, and human error. At first, the result looked like a measurement problem. At what later records identify as Challenger Station 225, though some older sources list Station 215, the lead finally reached bottom at 4,475 fathoms. That was about 8,184 meters, or 26,850 feet. Everest could have been lowered into that water and still not risen above the surface. For the men on deck, the number did not arrive as a glowing revelation. It arrived as a change in tension. The line had stopped paying out. Somewhere below them, the weight had found the floor. They repeated the sounding. The second attempt mattered. If the line had caught on something, if the ship had drifted too far, or if the apparent bottom was only a false stop, the next reading should expose it. But the great depth remained. Repetition did not erase the error. It made the result more convincing. The crew had not seen the Mariana Trench. They had not entered its deepest depression. They had not photographed a creature, collected a mysterious object, or touched a landscape with their hands. They had measured an abyss indirectly, through a lead weight and a rope that had traveled more than 8 kilometers into darkness. The answer to what waited at the bottom was not an animal, it was a depth. The result changed the map because it gave the region a historical anchor. Later surveys would show that the deepest point lay still farther down, close to 11 kilometers, and that the trench contained several separate depressions rather than one perfectly defined hole. Modern multi-beam sonar would produce figures such as 10,984 meters plus or minus 25, while other surveys would cite 10,994 meters. Those later numbers came from sound, computers, corrections, and repeated passes. The original discovery began with a man watching line disappear. The name remained. The deepest basin became Challenger Deep, named for the ship that first revealed this extraordinary depth and later reinforced by the name of HMS Challenger 2. The vessel had not found the final lowest point. It had found a section of ocean deep enough to send every assumption back onto the deck for inspection. For a crew trying to determine whether they had found an error or a genuine abyss, the answer was written twice in the log. The line had not failed. It had reached the bottom. Number 12. 20 minutes at the bottom. On January 23, 1960, Don Walsh and Jacques Piccard were already disappearing from the world. Inside the Trieste steel pressure sphere, there was no room to stand and no way to open a door. Above them floated a buoyancy tank carrying 22,000 gallons of aviation gasoline. Its volume would later help lift the craft back to the surface, but during the descent, it was only part of a machine sinking under its own weight. The USS Wandang had towed them over Challenger Deep, near Guam. Now the tug was receding above them, and the Trieste was descending toward an ocean nearly 7 miles deep. It was not a submarine in the familiar sense. There were no corridors, no control room, and no safe route out. The sphere was a small metal capsule surrounded by water whose pressure would rise toward nearly 1,000 atmospheres. At that level, the survival margin was not measured in comfort or convenience. It was the steel shell, the seals, and the systems that could still bring them home. Iron shot, held by electromagnets, served as ballast. Release the shot, and the bathyscaphe would rise. Lose power, and the ballast would fall away automatically. That was the plan. At depth, plans became very small. Walsh and Piccard descended for almost 5 hours. The surface light faded first. Then the blue disappeared. Outside the sphere, the ocean became black, and the last familiar reference points vanished. At roughly 30,000 feet, a sharp crack broke through the sphere. One of the viewing windows had fractured. For a moment, the entire dive narrowed to that sound. The window was tiny, but it was their only direct view of the outside world. Beyond it waited water under immense pressure. Inside it were 2 men who could not repair the damage, inspect the exterior, or leave the sphere if the fracture spread. The inner pressure seal held. They continued. The descent that rank as number 12 in Trieste's series now became a decision with no comfortable answer. Aborting would require releasing ballast and beginning the return with the damaged pane beside them. Continuing might let the crack propagate before they reached the bottom. They had no reliable way to know which danger was growing faster. Their communication with the surface was limited, and every minute spent evaluating the window was a minute added to the descent and the eventual ascent. The sphere was still moving downward, but the mission had changed. Reaching Challenger Deep was no longer enough. They had to decide whether the craft could survive staying there and then survive coming back. The Trieste sank deeper. The damaged pane remained beside their faces as a warning. There was no way to isolate it from the cabin, and no second view through which to check what the fracture had hidden. Instruments told them how far they had gone. The window could tell them what was outside if it remained intact. Then, after nearly 5 hours, the bathyscaphe touched the bottom. The depth was reported at about 35,800 feet, with official figures varying slightly around 35,800 to 35,814. That distance meant a long ascent, even if every release mechanism worked. At the bottom, the arrival was almost quiet. Trieste settled into the seafloor, and the view disappeared in a cloud of disturbed sediment. First human visit to Challenger Deep had reached its destination, but the damaged window had reduced the time available to inspect it. Walsh and Piccard had only about 20 minutes. Visibility was poor, and the cracked pane left them uncertain how much of the outside scene they could trust. They waited for the cloud to thin, knowing that remaining longer increased the value of the observation, while also extending the return journey with a compromised window. Somewhere beyond the suspended particles was the floor, measured by soundings and imagined from maps. Now it was close enough to touch, but almost impossible to see. Piccard later reported seeing what looked like a flatfish moving across the bottom. The claim became one of the most famous observations in deep ocean history. It also became one of the most disputed. True bony fish were not expected to survive at that pressure. The animal may have been an invertebrate, perhaps a sea cucumber. It may have been a shape distorted by the clouded view and the narrow, damaged window. The Trieste had no camera system capable of recording every movement. It had 2 observers, a short deadline, and a single uncertain glimpse through a pane they already feared. The sighting could not be revisited from the sphere, because any attempt to stay longer increased the risk attached to their only rude home. The 20 minutes ended. Walsh and Picard released the iron ballast. The electromagnets let go. The weight fell away, and the Trieste began its long climb toward daylight. The mechanism that had carried them down now became their way out. For hours, the damaged window remained with them. 22,000 gallons of gasoline in the buoyancy tank helped lift the craft, but ascent still depended on the pressure sphere, the ballast release, and the fracture remaining stable. A failure would not merely end the observation. It would leave the men at a depth where rescue was impossible. When the Trieste finally returned to the surface, Challenger Deep had given them no giant creature and no mysterious artifact. It had given them a cracked window and a possible flatfish that science could not confidently identify. The men had not simply measured the abyss from above. They had entered it, watched their only direct view become unreliable, and left before the evidence could be secured. The window had threatened the mission, and the sighting had outlasted the moment that produced it. But what had Walsh and Picard actually brought back? Evidence, illusion, or the first warning that later dives would face a much harder problem than darkness alone? Number 11. The floor that looked alive. At the bottom of Challenger Deep, the view did not reveal bare rock. It turned brown. On January 23, 1960, Jacques Picard watched through the small acrylic viewport of the bathyscaphe Trieste as the seafloor rose toward him. Lieutenant Don Walsh called out the shrinking distance from the fathometer. The sphere descended toward 10,900 meters, nearly 35,800 feet below the surface. Then the craft touched down. A pale cloud burst upward. It spread across the window like underwater smoke. Fine particles hung in the weak light before settling back onto the floor. The deepest place on Earth disappeared inside its own sediment. The first humans to reach it were not looking at a desert of black stone. They were looking at a soft brown layer disturbed by their landing. What exactly were they seeing? Walsh is often quoted as describing the material as diatomaceous ooze, but that wording should be treated cautiously. It may preserve his observation, or it may reflect a later interpretation of the cloud's appearance and likely composition. The original report was more limited. A soft brown bottom and fine material rising around the sphere. That distinction matters because the cloud could not identify itself through the viewport. Dead plankton sink from the sunlit ocean. Shell fragments descend with them. Organic matter is consumed, scattered, and altered on the way down. In a trench this deep, some of that material can accumulate, be compressed, and remain in the sediment. But Picard and Walsh could see only particles moving in the landing cloud, not the organisms that had produced them or the layers beneath the floor. The cloud may have contained remains of life that once lived thousands of meters above the trench. It may also have included ordinary fine sediment displaced by the impact. Without a core from the exact touchdown point, the iconic first human observation could not establish what was actually present there before Trieste arrived. Later researchers examined Mariana Trench sediment in greater detail. A core identified as JL7KGCO5 contained diatomaceous material near the sediment surface, including fragments associated with the large diatom Ethmodiscus rex. These were not large animals moving across the floor. They were broken microscopic shells, preserved after their owners had vanished. That later evidence gives the brown cloud a plausible connection to the surface ocean, but it does not turn the 1960 descent into a direct sample return. The biological signal was in particles that had sunk, accumulated, and survived burial. Trieste disturbed those particles before anyone could determine their proportions at the landing site. The trench floor was therefore connected to events far above it. Surface productivity, transport through the water column, and older changes in ocean conditions could all leave traces in the sediment. To Picard and Walsh, those traces were hidden inside the material obscuring the window. They could not see a paleoceanographic record. They could see only the physical evidence that something had settled there and had been lifted again. The uncertainty had a concrete consequence. The first visual account from Challenger Deep could describe the seafloor's texture, but it could not prove its composition. Such as diatomaceous ooze linked the landing site to microscopic life in the upper ocean, yet no sealed container came back from the exact point where the sphere touched down. No laboratory could immediately separate diatom fragments from ordinary trench sediment. The official record remained narrower than the phrase. It documented a soft, brown bottom and a cloud of fine material. Later sediment studies showed that Mariana deposits can preserve biological and chemical signals from the ocean above, including traces of changing productivity, transport, and climate. Those studies explain why the interpretation is reasonable. They do not provide the missing sample from January 23, 1960. You are looking through Picard's window. The view is fading. The particles are moving. There is no second look, no close-up, no instrument waiting outside the sphere to identify them. What seemed like an answer was still only a label placed on a cloud. And the cloud may have erased the evidence it was supposed to reveal. The first visual record of Challenger Deep was permanently contaminated by the landing itself. Number 10. Life without sunlight. On March 2, 1996, the Japanese remotely operated vehicle Kaiko reached the bottom of Challenger Deep. It was 3 meters long and descended nearly 10,898 meters below the surface. Its touchdown was recorded at approximately 11 degrees 21.111 minutes north, 142 degrees 25.949 minutes east. There was no sunrise down there, no usable sunlight, no ordinary food chain beginning with plants, only mud. Kaiko brought that mud back to the laboratory. The question was brutally simple. What feeds organisms where sunlight never arrives? The first answer was not a fish, an amphipod, or anything large enough for a camera to notice. It was hidden between sediment grains. Researchers recovered bacterial material from the sample, including actinomycetes, fungi, ordinary non-extremophilic bacteria, and organisms adapted to cold or alkaline conditions. 2 strains mattered most. They were obligately barophilic. Pressure was not merely something they survived. It was part of the environment they required. Remove enough of it, and the organisms stopped behaving like deep-sea specialists. Their biology had been tuned to a world where the water above them weighed roughly 10,000 meters. Number 10 is the ecosystem that runs without a sunrise. The distinction sounds technical, but it changes what the mud means. A cell can sink from shallow water. A fragment can be transported. Contamination is always possible when a sample travels from the crushing seafloor to a ship, then into a laboratory operating at ordinary pressure. Kaiko's sample contains something harder to dismiss: microbes whose best growth occurred under extreme pressure. An isolate, identified as DB21MT-2, grew most effectively at 70 megapascals. Another, DB21MT-5, preferred 80 megapascals. That is about 700 to 800 times atmospheric pressure at sea level. At the surface, the pressure would fall so sharply that the cell's membranes, enzymes, and internal chemistry would no longer be operating in their natural range. At Challenger Deep, those same conditions were closer to home. The samples suggested more than isolated names on a culture sheet. Filamentous structures observed later at roughly 10,677 meters were cautiously interpreted as a microbial mat community. Not a broad green carpet, but a mesh of microscopic growth spread through the upper layers of mud. The visual evidence was limited. The decisive encounter came when the isolates were placed into controlled conditions and the pressure was changed. At 70 megapascals, one strain reached its preferred range. At 80 megapascals, the other did the same. The response was repeatable enough to turn a visual suspicion into a physiological result. The trench was not merely collecting dead material. Its sediment was sustaining a community whose growth depended on the very pressure that makes the site so hostile to surface life. That chemistry had to come from somewhere. No sunlight reaches the floor, so there is no photosynthesis at the source. Energy can be released through chemical reactions involving compounds in the sediment and water. Microbes can use reactions involving carbon, sulfur, nitrogen, hydrogen, and other chemical gradients. Organic matter also sinks from above: particles, fragments, and microscopic remains that survive the journey down. The ocean supplies the raw material. The trench supplies the pressure. At the bottom, life does not need a hidden monster to remain active. It needs energy and forms a surface ecosystem would barely recognize. You are no longer looking through a vehicle window. You are looking at a sealed sample under laboratory pressure, where the mud has been separated, cultured, and tested. A living organism is multiplying because the pressure is high enough. Lower the pressure, and the evidence changes. That does not prove every cell in the original sediment belonged to a permanent native community. Sampling and transport can distort the picture. Culturing favors organisms that grow under laboratory conditions, while countless others remain invisible. The exact structure of the original microbial mat is still partly inferred. The pressure-loving physiology is not an inference, it was measured. And that measurement sharpens the consequence. Abyssal life may not be a thin residue of surface biology carried downward and barely surviving. At least some of it is organized around conditions that would destroy its surface relatives. The deepest mud may represent a separate biological regime, one defined not by sunlight, but by pressure, chemical gradients, and slow burial. The next question is harder. How much of the trench's hidden ecosystem depends on organisms that cannot survive the journey to the surface, and what else is active in the sediment that no culture can reveal? Number 9. The armored scavenger. The scavenger wearing armor made from the seafloor. In 2009, a Japanese team brought small, pale crustaceans up from Challenger Deep, roughly 10,900 meters below the surface. The animals were about 2 inches long. Their bodies were narrow, segmented, and almost translucent under the laboratory lights. They looked like shrimp designed by a machine that had never seen the sun. They were Hirondellea gigas, giant amphipods. They were not rare visitors passing through. Around 11,000 meters down, they are dominant residents of the trench floor. Organic material falls from above. The amphipods find it, tear it apart, and turn what remains into energy. Ordinary scavenging is only the first clue. The central puzzle is how an amphipod keeps its shell from dissolving in water rich in dissolved metals, where the pressure is close to 1,000 times that at the surface. The animal needs a hard exoskeleton for protection. The chemistry of the trench should make that armor unstable. Scientists had to look inside the body for the answer. In specimens examined by Hiroyuki Kobayashi and his colleagues, scientists found an enzyme called HG-cell. It breaks down cellulose, the tough material that gives wood its structure. The reaction produced glucose and cellobiose at roughly a 2-to-1 ratio. The animal can process plant debris, not a leaf drifting past a coral reef, not algae scraped from sunlit rocks. Wood that began in a forest, on a ship, or along a coast, then sank through kilometers of water and settled in one of the darkest places on Earth. The trench does not receive a complete meal. It receives fragments, a splinter, a fiber, a buried piece of carbon that has traveled farther than most living things ever will. Inside the amphipod, HG-cell opens that material again. The deepest ecosystem is often imagined as a place powered only by carcasses and microscopic sludge. Hirondellea gigas carries a biochemical link to the surface world. Some of the carbon-feeding life at Challenger Deep may have started as wood beneath sunlight, long before it became food beneath crushing pressure. The enzyme proves capacity, not a full menu. It does not tell us how much of the animal's diet is wood, how much is carrion, how much is microbial material, or how much is everything else that settles into the mud. It does reveal a body built for scarcity. The armor is stranger still. Researchers studying the species found aluminum-rich biological protection associated with the exoskeleton. The amphipod extracts aluminum from its surroundings and uses it to stabilize the shell. The process begins in the gut, where aluminum from sediment and seawater binds to organic compounds. Those compounds move toward the exoskeleton and form a protective layer that helps prevent the shell from breaking down. The animal is manufacturing armor from the chemical environment that could destroy it. The raw material arrives with the food. Woodfall, sediment, metals, and organic compounds enter the same small digestive system. One process breaks cellulose into usable sugars. Another helps turn dissolved aluminum into structural protection. The gut serves as a kitchen, refinery, and armor workshop. A second material now reaches the bottom, though no hadal animal evolved to use it. January 29, 2017, scientists examined 10 Hirondellea gigas collected from 10,890 meters in the Mariana Trench. Every one contained synthetic or semi-synthetic fibers. Some included low-density polyethylene, the plastic used in packaging and countless disposable products. All 10. The sample was small, but the result was clear. The same scavenger capable of converting wood from the surface world into food had also swallowed fibers manufactured by the surface world. The fibers did not need to be carried directly to Challenger Deep by a single current. Plastic can fragment, sink, attach to organic particles, and move through the water column over time. Around the Pacific, similar contamination has been recorded in amphipods from 6 hadal trenches. The animal's gut preserves 2 routes downward. One begins with a tree, the other begins with a factory. Both arrive as debris. Both are picked up by a scavenger moving across sediment that has never seen daylight. Their chemistry is vastly different, but at the bottom, they occupy the same narrow digestive chamber. A creature whose shell survives by extracting protection from the seafloor may feed on carbon from forests and fibers from plastic bags. It is adapted to the trench, yet its stomach records the planet above it. The question is no longer whether life can function at 10,994 meters. Hirondellea gigas has answered that with an enzyme, a shell, and a working digestive tract. What remains unclear is how much of that system is natural and how much contamination is now being folded into it. Inside the gut, organic compounds bind the metal. The metal stabilizes the exoskeleton. The scavenger moves across the trench floor wearing armor made from the seafloor. In the laboratory dish, one amphipod lay motionless beside a strand of polyethylene, its pale shell carrying the chemistry of the trench. Number 8. The vacuum cleaner on legs. At first, the seafloor looks empty. Then a translucent shape rises from the mud on a set of thin, awkward legs. It does not swim or chase. It walks across the darkness, gathering whatever the ocean has allowed to settle. This animal survives by eating the ocean's leftovers. On May 28, 2009, biologist Tim Shank and the Woods Hole team sent the hybrid robot Nereus into the Mariana Trench. At 8,984 meters, its manipulator reached into the sediment and brought back something that looked less like a sea cucumber than a fragment of tissue filled with mud. The animal belonged to the genus Scotoplanes, the sea pig. Elongated tube feet ran beneath its body, while feeding structures helped it work through soft sediment. It may even have represented a species new to science. The name sounds almost cheerful for a place like this. The animal, however, fits the trench floor precisely. It has no jaws built for tearing, no armor for fighting, and no sudden burst of speed. Its body is soft, its underside broad, and its legs keep it moving across ground where almost everything arrives from above. The deep ocean is not empty. It receives dead plankton, tissue fragments, drifting organic particles, and anything else that survives the long fall from sunlight into darkness. The sea pig does not hunt large prey. It collects the edible fraction from the sediment, one mouthful at a time, turning material left by the ocean into usable food. The Nereus specimen was not the strangest sight recorded at the bottom. On March 26, 2012, filmmaker James Cameron reached Challenger Deep inside Deep Sea Challenger. The descent took 156 minutes. Through the viewport, the silty plain appeared almost lifeless. No obvious swarms moved across it. No large animals crossed the frame. Nothing announced itself. Then Scripps researcher Natalia Gallo examined the video again. She went through it frame by frame. A slight disturbance appeared in the sediment. Then another. Small gray shapes emerged from the darkness, semi-transparent and almost motionless. They were holothurians, sea cucumbers living between 10,876 and 10,908 meters. In 2-minute video samples, Gallo's team counted 65 individuals. They had been there the whole time. The animals extended 3 to 5 oral tentacles into the water and mud, yet left almost no trails. There were no obvious fecal coils and no dramatic crawling. Against the brown sediment, their bodies were nearly invisible. Cameron had described the bottom as showing literally no sign of life. The recording contained dozens of animals moving through the frame like pieces of the seafloor itself. One detail made the scene look organized. Whenever several animals appeared together, they pointed in the same direction. They were not scattered randomly or facing one another. They were aligned. Gallo's team proposed that a current was delivering edible particles toward their tentacles. The animals may have been positioned like small biological filters, each facing the same invisible flow. Their stillness was not inactivity, it was placement. The trench supplied the food. The animals only had to stand where it arrived. Their exact identity remained uncertain. The 2009 animal was assigned to scotoplanes. The Challenger deep population was tentatively placed in Peniagone, a related Elpidiid lineage. Animals shared the same pale, pig-like appearance, but sea pig is not a universal name for every hadal sea cucumber. Without an intact specimen, the Challenger deep animals could not be formally named. Their identity was missing, but their abundance was not. In trawls deeper than 7,500 meters, holothurians have sometimes made up between 75 and 98% of the animals recovered. In some samples, they exceeded 90% of the biomass. Researchers have called such places a kingdom of holothuroidea because these soft-bodied deposit feeders dominate the material brought back from the deepest habitats. Yet scotoplanes is normally recorded from roughly 1,000 to 6,000 meters and can reach about 17 centimeters in length. The animal collected by Nereus at 8,984 meters was far outside its usual recorded range. The animals filmed near Challenger Deep may belong to something different again. The opening image now looks different. You are not watching a strange creature wander through empty mud. You are watching an animal find the trench's invisible delivery system, extend its tentacles, and collect organic particles arriving from the ocean above. Its legs carry it over the sediment. The current sets the direction. Its mouth takes what remains. The footage ends before the answer appears. A gray body, 3 tentacles, a patch of disturbed silt, several animals facing one direction. No specimen, no confirmed species, no direct proof of whether they scraped particles from the mud, filtered them from the current, or switched between both. The animal was visible, but its name was not. Its food was present, but its method remained hidden. On the screen, one sea cucumber moved across the sediment, stopped, and extended its tentacles into the flow. Then the frame ended. Number 7. The fish that stops above the bottom. In November 2014, baited landers descended into the Mariana Trench near Guam, sinking for hours through darkness toward nearly 8,000 meters. Cameras watched the traps settle. Amphipods rushed across the bait. Then a pale fish drifted into view. It had translucent skin, a broad head, and a tapering body that looked almost unfinished. No armor, no enormous jaws. Just a soft, gelatinous animal hovering calmly above the sediment, while pressure approaching 800 times that at the surface pressed against every part of it. The old image of the deepest ocean had suggested a hard limit. Fish belonged to the upper world. The hadal zone belonged to crustaceans, microbes, and soft-bodied scavengers. Yet here was a vertebrate moving through water deeper than any familiar fish should survive. The observation established an extreme depth, not the absolute bottom. The animals collected during those missions came from between 6,898 and 7,966 meters, while earlier observations had placed the Mariana snailfish as deep as about 8,076 meters. Challenger Deep reaches approximately 10,935 meters, or 35,876 feet. Almost 3 vertical kilometers separated the known fish from the deepest seafloor. That gap was not merely a missing measurement. It marked a pressure increase large enough to challenge the chemistry that keeps a vertebrate alive. In 2017, Mackenzie E. Geringer and Thomas D. Lindley formally described the species from 37 collected individuals. They named it Pseudoliparis swirei, honoring Herbert Swire, an officer aboard HMS Challenger. The name linked the fish to the expedition that first established the existence of the trench system, but the animal itself answered a more immediate question. How can a vertebrate function under such pressure? Its tissues are loose and water-rich. It has no large gas-filled swim bladder to collapse. Its body yields rather than opposing the surrounding water, reducing the structural stresses that would damage a rigid form. At around 8,000 meters, that design allows the fish to hover over sediment, turn toward bait, and move through the water column with little wasted effort. But pressure does more than squeeze a body. It alters the shape and motion of molecules. Protein folds become less stable, so enzymes can lose the precise structures required for metabolism. cell membranes become less fluid, slowing the movement of nutrients, ions, and electrical signals across them. Nerve cells depend on those signals to coordinate movement, while sensory organs depend on them to detect chemical and mechanical cues. If compensation fails, muscles no longer receive reliable instructions, cells cannot maintain their internal chemistry, and the animal loses the ability to feed, orient, or control its body. The extra 3 kilometers would add hundreds of atmospheres of pressure. A flexible body could absorb some mechanical compression, but it could not indefinitely prevent pressure from disrupting molecular reactions throughout every cell. Critical failure would therefore be biochemical before it became visibly structural. Metabolism would slow or lose coordination. Membranes would stop supporting normal nerve function, and the systems that make a moving vertebrate possible would collapse together. The deeper the lander goes, the less certain the picture becomes. A fish filmed at 8,000 meters is not evidence of fish at the bottom, and a camera that sees no fish is not proof that none are there. The final range may be physiologically impossible for vertebrates, or the record may end because expeditions have not yet sampled the right place. In 2023, researchers filmed a different, undescribed Pseudoliparis snailfish at 8,336 meters in the Izu-Ogasawara Trench. That observation extended the known range for a filmed fish, but it also sharpened the question. The fish was still far above Challenger Deep, and the failure boundary remained unobserved. Officially, Pseudoliparis swirei remains a hadal fish, known from the upper part of the Mariana Trench, thriving around 8,000 meters near Guam. Its body plan reveals how vertebrate life can enter the hadal zone, but not how far that strategy can continue. Below the observed range, the next descent would have to find whether a living fish is absent because no vertebrate physiology can cross the threshold, or because the exact boundary has not yet been reached. The next lander will not simply be looking for another record. It will be looking for the depth at which the chemistry of a vertebrate body finally stops working. Number 6. The giant single cell, the creature whose entire body is one cell. In July 2011, Scripps microbiologist Doug Bartlett watched an untethered camera descend into the Serena Deep. Its lights reached a place where one small mistake could destroy the machine. The drop cam fell alone through more than 10,000 meters of water. No pilot, no tether, no second chance. At the bottom, it filmed pale shapes scattered across the sediment. They looked like small sponges, or pieces of torn fabric, but they were alive. They were xenophyophores, giant single-celled organisms built from sediment grains, organic particles, and material gathered from the seafloor. Some measured several centimeters across. A few approached 10 centimeters, roughly 4 inches. That is enormous for one cell. Most cells need a microscope. This one could be seen by a camera operating almost 6.6 miles below the surface. The discovery pushed their confirmed depth range far beyond the previous record of about 7,500 meters in the New Hebrides Trench. The deepest parts of the ocean were not simply holding microscopic life in the mud. They were supporting single cells large enough to shape the landscape around them. The contradiction was immediate. How can one cell become that large without losing control of its own chemistry? How can a single unit remain stable under pressure hundreds of times greater than at the surface, in darkness, cold, and almost permanent hunger? Imagine a normal cell expanded to the size of a coin. Its interior would become a logistical disaster. Nutrients would have farther to travel. Waste would have farther to leave. Signals would slow. The cell should become less coordinated as it grows. Xenophyophores avoid that problem by looking less like ordinary cells and more like living construction sites. Their bodies are not compact spheres. They are branching, delicate networks spread through the upper sediment. The organism extends a living mass through tubes and chambers. This increases its contact with the surrounding environment without creating one dense interior. The trench floor is not a solid platform. It is soft brown sediment, made from material that has settled out of the water above. Organic particles arrive slowly. Microbes and scavengers consume some of them. Others are buried. A xenophyophore gathers this loose material into a structure that can persist on the seabed. Its body becomes part organism, part archive. The grains it collects record the local environment, mineral fragments, organic remains, and particles carried down through the water column. A xenophyophore does not merely live on the trench floor. It incorporates the trench floor into itself. Its size creates another effect. Lisa Levin and other deep-sea biologists found that xenophyophore structures can host diverse multicellular organisms and microbes. Their fragile branches create surfaces, pockets, and shelter on an otherwise open plain. The sediment shifts beneath every small animal. In that setting, even a few centimeters of structure can matter. A single cell can become a tiny reef. Not a reef made of stone. A reef made of mucus, sediment, and time. You are lying on the floor of the Serena Deep. There is no sunlight, no familiar horizon, and no hard ground beneath you. Around you, the sediment looks almost empty, and the camera lights reveal a pale network resting across the mud. It does not move like an animal. It is not rooted like a plant. Yet it has created one of the few visible pieces of architecture in the entire landscape. The trench is not waiting for a single monster to appear. Life is assembling itself from small advantages. A body that tolerates pressure, a structure that catches food, and a surface that gives other organisms somewhere to settle. The 2011 cameras did not solve the deepest mystery. Xenophyophores have been reported from extreme depths, and sightings have been associated with the deepest parts of the Mariana Trench. The best confirmed record remains the Serena Deep observation at 10,641 meters. The absolute bottom of Challenger Deep, around 36,000 feet below the surface, has not yielded a complete inventory of stable xenophyophore populations. The map remains incomplete. A camera can record a shape, but it cannot always reveal whether that shape is a thriving population, a temporary growth, or a fragile structure carried into place by the sediment. The answer to the size paradox is hidden inside the organism itself. Xenophyophores are not giant versions of ordinary compact cells. They are sprawling internal networks, supported by sediment frameworks and built from the environment around them. Their apparent mass is partly living material and partly collected architecture. The one cell stays large because it distributes itself through a structure that turns the seafloor into scaffolding. The deepest single cell is not a swollen speck. It is a living landscape. 10,641 meters. The ocean floor was already being engineered by an organism too large to see as a normal cell, but too simple to divide into anything more familiar. As one scientist put it, these creatures can be ecosystem engineers of the deep sea. Number 5. Bodies built for pressure. In 2016, a NOAA camera hangs in black water over the Mariana region when something pale drifts into the lights and almost disappears inside them. No shell, no spine, no obvious face. Just a body with edges so thin the ocean seems to pass through it. The vehicle sees it for a few seconds, then it is gone. Back on the screen, what remains is a question with tentacles. The bodies that survive by giving up their bones. NOAA defines the hadal zone as the band from 6,000 to 11,000 meters, and Challenger Deep is its best-known address. At that depth, the ocean stops looking ordinary and starts acting like a test chamber. It is dark, near freezing, chemically uneven. The pressure is crushing. For a long time, the picture of survival down there leaned toward armor, compact muscle, hard parts, anything built to resist. Some of the strangest residents take the opposite approach. They stay soft. In 2016, the Okeanos explorer spent 59 days surveying the Marianas Trench Marine National Monument and nearby waters. 22 dives, hours of live video. Baseline work in one of the least mapped biological spaces on Earth. Among the hardest animals to fit into that baseline were the gelatinous ones. Jellyfish, comb jellies, salps, and other drifters whose bodies are mostly water and whose lack of hard parts makes them notoriously easy to lose. A net can come back looking empty even when the water was not. A rigid fish, a crustacean, a shell-bearing animal. Those usually leave something recognizable behind. A gelatinous animal can tear, collapse, smear, or vanish into a sample as if it had never been there. Cameras changed the record more than tow lines did. The trench did not suddenly fill with jellies because they arrived late. It started filling with jellies on screen because we finally had a way to catch them without touching them. The logic becomes clear as soon as you stop expecting a deep animal to be built like a submarine. Gas-filled spaces are a problem under extreme pressure. Hard frameworks can become liabilities if the whole body has to tolerate compression. A body that is mostly water gives the pressure less to crush in the first place. It does not beat the hadal environment. It slips around part of the problem. Some of the trench's most elusive forms look less designed than half erased. A comb jelly can appear as a floating seam in the water. A salp can look like a transparent tube with no business functioning at all. A hydromedusa may show a bell, trailing structures, a pulse of motion, and then vanish before it leaves anything clean enough for a name. Deep-sea observations are often brief and partial, made in darkness. The vehicle turns, the animal folds, marine snow crosses the lens. Identification becomes forensic work from a handful of frames. Those frames keep pushing gelatinous life deeper. A 2023 review of gelatinous records pointed to a predatory tunicate observed in the Mariana Trench at 7,799 meters. Not a jellyfish in the strict sense, and that matters here, but still a soft-bodied gelatinous hunter recorded well inside hadal territory. Elsewhere in the Western Pacific, the pattern goes deeper still. A Trachymedusa record in the Philippine Trench reached 10,063 meters, pushing one hydrozoan lineage beyond 10,000 meters. Benthic ctenophore in the Kermadec Trench was documented at 10,040 meters. Different trench. Different animal. The same pattern. Soft-bodied life was reaching deeper than older sampling methods had shown. This is not a story of crowds. The frame does not fill with swarms. You get rare, ghost-like appearances. A shape in the lights. A bell with no clear species name attached. A drifting predator seen once at a depth where almost everything we know still comes in fragments. Expedition after expedition, the trench yields something slightly different, and gelatinous animals are part of the reason. They are easy to miss, hard to preserve, and often too incomplete on video to classify with confidence. The unidentified animal in the lights remains what it looked like from the start. Evidence before explanation. Some things can still be said. If it moves with a pulsing bell, trails feeding structures, or shows the body plan of a tunicate or medusa, the footage can narrow the field. It was living there in situ, not crushed in a net on the way up. At hadal depth, a soft body is not a defect. It is one workable answer. Modern vehicles have also shown that gelatinous lineages reach far deeper than many records once suggested. Video alone often stops short of a full identity. Not always species. Sometimes not even the level of certainty a biologist would want. No hard parts. No specimen in hand. No genetic sample. Just a luminous body crossing the lamps, visible only because a machine happened to be there when it passed. So the opening sight resolves only part way. Translucent. Gelatinous. Most certainly one of the soft-bodied drifters or hunters that modern cameras have finally begun to catch in place. And still unresolved in the one way a catalog cares about most. What exactly it was when the lights found it and it slipped back out of frame. Number 4. The ambush fixed to the wall. In August 2022, the Full Ocean Depth Submersible DSV limiting factor was moving along the Western Pacific Trench system when its cameras picked out something that did not behave like drifting jelly life at all. It was fixed in place. A single stalked animal stood off the wall at roughly 7,799 meters in the Mariana Trench, pale against the black water, waiting. At first glance, it looked harmless, in the way sea squirts often do. A bag of tissue, a tube on a stem. The kind of body plan most people would file under filter feeder and never think about again. But the footage created a very specific problem. Was this animal simply straining particles from the water, the deep-sea equivalent of a quiet pump? Or was it doing something much more direct? The answer mattered because the trench keeps breaking the same assumption from different angles. If the deepest place on Earth is dark, cold, and under crushing pressure, you expect fewer moving parts, fewer specialized tricks, fewer ambush hunters nailed to the canyon wall. Instead, the camera found a body built around an opening that looked less like a sieve and more like a mouth held open on purpose. The 2023 paper by Alan J. Jameson, Dougal J. Lindsay, and Hiroshi Kitazato treated the sighting carefully, but the shape tells the story. This tunicate belonged to the family Octocnemidae, a group known from the deep sea. Its oral siphon was enlarged into a broad hood, not a narrow intake, not a passive little nozzle, a hood. In related predatory tunicates, such as Megalodicopia hyans, that hypertrophied opening functions as a trap, able to catch small crustaceans and other passing animals. Water still moves through the body, but the architecture changes the job completely. The feeding apparatus is no longer just collecting suspended bits. It is intercepting prey. You can see why that distinction took time to settle. Sea squirts have a reputation problem. On piers and reefs, they are the wallflowers of marine biology, attached animals that mostly sit there filtering. But down here, the familiar outline has been repurposed. Deep canyon walls offer hard surfaces that the soft trench floor does not, so an anchored animal can hold position above the sediment and face the traffic drifting past. Same basic lineage, very different behavior. And this one was not a fluke glimpse. The researchers recorded this kind of tunicate 9 times in the Mariana Trench during the Ring of Fire expedition. 9 is not a swarm. 9 is not abundance. But 9 separate observations are enough to move the animal out of the category of accident and into the category of presence. Then the expedition found the same kind of tunicate again at 8,077 meters in the Izu-Ogasawara Trench. That single extension pushed the known lower depth limit for tunicates down by 1,726 meters. So somewhere in the middle of all these trench organisms that either crawl through mud, drift through darkness, or scavenge what falls from above, you get the harmless-looking trap anchored to the canyon wall. That is the image the document leaves behind. Not a grazer, not a sponge, a sit-and-wait predator with its hood open in water cold enough to numb machinery. There is a dry forensic logic to that claim. Start with the opening. A broad oral hood increases encounter area. Then the posture. Elevated on a stalk, it projects into the passing flow instead of burying itself in the detritus. Then the relatives. Predatory members of this deep-sea group are already known, and their oversized siphons are associated with prey capture, not simple suspension feeding. Then the behavior in situ. Fixed to the substrate, mouth held as an ambush chamber, exactly where small crustaceans or other drifting animals could be funneled in. Piece by piece, the witness statement stops sounding like sea squirt and starts sounding like trap. But the same record that confirms the predator also limits it. The Mariana Trench is not one surface. It is walls, slopes, benches, sediment plains, and finally, the deepest floor. A vertical habitat can host animals the flat bottom does not. So when later viewers hear that a predatory tunicate was found in the Mariana Trench, there is an easy mistake waiting to happen. The footage does not place it at the absolute bottom. At Challenger Deep, filmed at 10,908 meters, the visible megafaunal community looked different. Sea cucumbers, amphipods, xenophyophores. Those are the animals reported from that first community analysis, not tunicates standing like snares on stalks. Even the revised deepest seafloor measurements, around 10,935 meters with only a few meters of uncertainty, do not help the tunicate claim. The point is not whether the trench is deep. The point is where inside that depth this predator has actually been seen. That narrowing matters because trench discoveries are often introduced as if the whole abyss were one room. It is not. Species found at 7,799 meters in the Mariana system is part of the trench story, but not proof it occupies the lowest floor of Challenger Deep. The paper is explicit on that boundary, and it has to be. Sampling effort drops hard with depth. A few sightings can reveal a range extension, but they do not tell you population size, breeding sites, prey choice, or whether these are residents spread along hadal slopes or isolated individuals near the upper margins. So the first third question does get answered. This organism was not merely filtering water in the ordinary sea squirt sense. Its feeding structure and observed posture identify it as an active ambush form, a predatory tunicate built to catch passing animals. But the second question stays open, and it is the one the trench keeps handing back. How far down does this hunter really go? The cleanest line in the record is still the most limiting one. Challenger Deep's filmed megafauna, at 10,908 meters, consisted of elpidiid holothurians, amphipods, and xenophyophores. Number 3. The light that gives you away. The signal that turns darkness into a hunting ground. On June 30, 2016, NOAA's Deep Discoverer moved through the Mariana Trench Marine National Monument. Not at Challenger Deep, but along the trench's dark slopes at roughly 2,500 meters. The cameras looked into a black water column where sunlight had vanished long before. Then a small, pale fish drifted into the beam, ghost-like, almost weightless. Noah later identified it as the first time a member of the Amphionidae family had been filmed alive in its natural habitat. That detail matters because the trench is more than deep. It is a place built around vision and light. At 11,000 meters, the pressure reaches about 8 tons per square inch, roughly 1,000 times sea level. But the visual game starts much higher. Between 200 and 1,000 meters, about 80% of ocean animals are bioluminescent. In a 2017 study, 76% of deep-water organisms observed off California showed the same ability, from the surface down to 3,900 meters. Down there, light is a working tool. It attracts prey, confuses predators, matches the faint glow from above, or warns something else away. The encounter with the cusk eel shows how quickly that tool can become a risk. The animal appeared only because the camera light reached it, and that same light may have altered its behavior, exposed it to larger hunters, or drawn other animals into the scene. In trench footage, the beam is never neutral. It can reveal a fish and also turn that fish into a target. In the trench system, every flash carries intent. A shrimp can burst out a luminous cloud. A jellyfish can flare when touched. Comb jelly can answer disturbance with a pulse of living light. Deep-sea fishes carry light organs that erase their silhouette from below. Some pandalid shrimp go further, releasing light from glands near the mouth and throwing out a glowing decoy in the dark. Darkness is not just something these animals endure. It is something they use. But the most important question is not what light can do in theory. It is what a live flash means in the trench right now. A burst on the slopes may be a predator closing in, a warning that an animal has been touched, or a signal meant for another creature just outside the frame. It may also be a response to the submersible itself. That is the danger of these encounters. The same flare that looks like a natural signal can be an alarm caused by observation. Living light and artificial light tell different stories, and sometimes they collide in the same frame. Noah's own descriptions lay out the logic plainly. Deep-sea animals use bioluminescence for predator evasion, prey attraction, prey detection, and communication. Not one purpose, several. That is why the trench can look empty and crowded at the same time. Most of the bodies are small. Most of the motion is brief. Most of the evidence arrives as a single flash inside a narrow cone of illumination. The live cusk eel off the Mariana slopes stands out for exactly that reason. It was not a glittering monster from the bottom. It was a tiny animal hanging in the water column where the beam reached it, visible inside a habitat full of creatures that either make light or react to it. In that environment, brightness is not spectacle. It is survival, and sometimes exposure. The water gets deeper, the evidence gets thinner. The strongest numbers come from surveys above the hadal zone, not from the trench floor itself. One point remains unresolved. How common is active bioluminescence below 6,000 meters in the Mariana Trench proper? And what happens if a vehicle's light triggers a chain reaction down there? The deepest verified footage still gives only fragments, flashes, behavior caught for an instant before the beam moves on. It proves life is there. It also leaves open a more urgent gap, whether the next light will reveal an animal or summon one from beyond the camera's reach. Number 2. The object that should not be there. On May 20, 1998, Kaiko's cameras settled over the floor of Challenger Deep and found a shape that did not move. No pulse of light, no drifting tentacles, just a pale sheet lying flat at about 10,898 meters, pressed into the sediment as if it belonged there. In a place where pressure destroys, cold slows, and darkness hides nearly everything, it still looked manufactured. The visitor that arrived without a submersible, Kaiko, was a Japanese remotely operated vehicle. Its operators were documenting one of the least accessible environments on Earth, not searching for pollution. The object entered the record by accident. A consumer product had reached full ocean depth without anyone carrying it there. That changed the trench from a remote boundary into a receiving point for material from the surface. On the monitor, the useful details were physical. The sheet was too thin for rock, too smooth for sediment, and too still for an animal. The camera could not identify its brand or route. It could establish something more consequential, an object made from minutes of use, had survived a journey into a place where direct observation is rare and removal is nearly impossible. For years, the image remained one entry in Japan's deep-sea observation records. Later, researchers at JAMSTEC combined thousands of dive observations in the deep-sea debris database. The wider pattern made the Kaiko finding harder to dismiss. In a 30-year analysis of deep-sea debris, 92% of recorded plastic items were single-use products, packaging, bags, and disposable plastic, rather than specialized hardware. A short, useful life did not prevent long-term burial. The important question is not whether the object looked like a bag. It is what that category proves. A single-use item on Challenger Deep means consumer waste can cross the ocean, descend through the water column, and enter the least surveyed part of the planet. The camera captured one item, but the finding broke the assumption that depth creates a barrier. The route remains unresolved. Waste may sink directly, travel through intermediate waters, move with currents from gyres, or be carried downward through biological processes. The uncertainty is not reassuring. It means there is no single pathway to monitor and no reliable travel time estimate. Human debris can reach the trench. The unknown is how many routes deliver it, and how frequently. The Kaiko image also makes the contamination look smaller than it is. One object, one frame, one piece of litter at the bottom of the world. The more serious evidence came without a visible object. In 2016, researchers collected sediment from Challenger Deep at 10,890 meters. Across the study's sampling sites, they extracted 32 microplastic particles from 3 kilograms of wet sediment. Converted to dry sediment abundance, the average was 71.1 particles per kilogram. Those figures are not merely a count. They show that the trench is not only receiving intact waste, it is storing fragments in the material that forms its floor. A large plastic sheet is an event a camera can notice. Microplastics are what remain after that event fragments, sinks, settles, and becomes part of the seabed. The object is visible contamination. Sediment is persistent contamination, potentially moving through organisms and food webs while becoming harder to locate or remove. That distinction changes the trench's role. Challenger Deep is not simply a place where rare debris has been photographed. It can function as a sink, a site where surface waste accumulates beyond routine inspection, then breaks down into particles that sampling may detect only after the original product has vanished. The broader numbers show why a single trench sample carries pressure. A review of 188 ROV dives in the Central and Western Pacific, spanning 150 to 6,000 meters, found marine debris on 17.5% of dives. This was not a census of Challenger Deep, but it showed how often debris appeared when researchers managed to look. The bottom is scattered evidence sampled through tiny windows. Every unsurveyed section is not empty, it is unmeasured. By 2020, global modeling estimated that between 3 and 11 million metric tons of plastic may already reside on the ocean floor. That figure cannot be assigned to Challenger Deep, but it changes the meaning of the Kaiko record. If billions of kilograms are settling below ordinary observation, then the deepest trench is not an isolated endpoint. It is one inaccessible compartment in a global storage system, where contamination can accumulate faster than expeditions can document it. The 1998 shape can be described as a plastic bag or sheet observed by Kaiko at 10,898 meters. But naming it does not close the case. It identifies the first visible edge of a process, surface waste reaching the trench, entering its sediment, and potentially disappearing from sight before anyone knows how much has arrived. The next object may not be found intact. It may already be part of the mud. Number 1. The question mark at 36,000 feet. A place can be measured to the nearest meter and still remain almost entirely unknown. In June 2020, Victor Vescovo descended into Challenger Deep aboard the submersible Limiting Factor. Instrumented landers waited in the darkness below. The target was the lowest seafloor on Earth. But the deeper the instruments looked, the less the phrase, "the bottom," seemed to mean. The deepest observed point was measured at 10,935 meters. It was not a perfect dot or a flat terminal basin. Ridges, sediment, slopes, and depressions spread across a trench that only a small number of vehicles have ever reached directly. The number became more precise. The place did not become complete. Challenger Deep is difficult to define as well as difficult to visit. Each survey can shift the apparent low point. Better instruments refine the depth, and the map changes with every descent. The most famous place in the ocean remains a moving target. Beneath that changing map, something is alive. The hadal zone begins around 6,000 meters and extends down to roughly 11,000. It is the ocean's pressure world. The water is near freezing. Sunlight is absent. Ordinary assumptions about animal life stop working. The fish found in the Mariana Trench live above Challenger Deep. Near the deepest zones, the visible community is dominated by microbes, amphipods, holothurians, and other invertebrates. Some of these organisms are familiar only because modern expeditions have finally carried baited traps, cameras, and genetic instruments into the darkness. Before 2014, much of what scientists knew about hadal life still rested on two major sampling campaigns from the 1950s, Galathea and Vitjaz. Those expeditions opened the subject, but they left enormous gaps between isolated specimens. A trap might recover an animal. A camera might record a few seconds of movement. DNA might show that something treated as familiar was genetically distinct. The same trench can still produce a species-level surprise in the 21st century. A Mariana Trench amphipod collected between 6,010 and 6,949 meters was described as Eurythenes plasticus in 2020. Its name reflected the human material found inside it. Genetic work later confirmed the hadal snailfish Pseudoliparis swirei, an animal adapted to depths around 8,000 meters. These discoveries do not turn Challenger Deep into a catalog. They show how much of the catalog is missing. The camera gives one kind of evidence. The trap gives another. Sediment gives another. DNA gives another. The methods do not necessarily describe the same community. Each one samples a different moment, depth, and patch of seafloor. The mystery is not whether life exists there. That has been proven. The mystery is how many forms of life can remain hidden inside a place that has already been visited, filmed, sampled, and named. The larger find lies in what has not been sampled, Challenger Deep itself. NOAA estimates that explorers have seen less than 0.001% of the deep ocean seafloor. Challenger Deep is only one small region within that immense dark floor. Even its deepest basin has been visited through narrow transects and isolated lander sites. A machine can cross the bottom, cannot inspect every hollow, slope, sediment layer, or chemical boundary. The documented evidence comes in fragments: a measured depth of 10,935 meters, a lander recording movement in the darkness, amphipods arriving at bait, microbial communities changing across sediment, an organism described from a trap and later reclassified through genetics, human debris resting where no human being could survive unaided. Each fragment is real. None is the whole picture. Imagine the descent ending. The lights of the limiting factor sweep across the seafloor. A shape appears at the edge of the frame. It is too distant to identify. The camera turns. The shape disappears into black water. The image could suggest a concealed creature. A quieter explanation fits better. It marks the boundary between observation and the absence of observation. The unknown thing at the bottom is not necessarily an animal hiding beyond the lights. It is everything the lights have not reached. Every new descent can redraw the map and the species list. One expedition finds a different organism because it brings a different trap. Another records a different patch because the seafloor is not uniform. A genetic sample reveals a separate lineage where the camera saw only a small body moving through silt. The trench has yielded proof of life, proof of contamination, and proof that depth is easier to state than to understand. It has not yielded a complete inventory. Challenger Deep is not an empty pit or a sealed chamber waiting to reveal one final monster. It is a living landscape sampled through pinholes. Its lowest point is still being refined. Inhabitants are still being sorted from names we know into species we have not yet recognized. What waits at the bottom? Enough life to prove the darkness is inhabited. Enough debris to prove the surface reaches it. Enough unmapped seafloor to make every answer incomplete. At 36,000 feet, the quietest thing is not the absence of a creature. It is the black water beyond the last recorded frame, where the boundary of knowledge begins.